Right. Now we are going to see the topic stem cell therapy under application of biotechnology. First of all, we must know what are stem cells. Stem cells are undifferentiated cells. They are not differentiated into any kind of cell. Nerve cell, blood cell, like this brain cells, they are not distinguished. They are undifferentiated cells which are found in most of our multicellular animals only. They maintain their undifferentiated state for even after numerous mitotic condition. How these stem cells are useful in the therapy treatment. For various treatment, how the stem cells in our human body they are useful, we are going to see. Right now, the stem cells, they are very important in the evolution of for the future of the medicine. In the future medical therapy, for the treatment of various genetic diseases and all, these stem cells are very useful. Right. What are stem cells? I said undifferentiated cells. They are not differentiated into nerve cell, blood cells like this. They are found in most of the multicellular animals like human being. Because for the treatment of human being only we are using the stem cell therapy. And they are having the characteristic features. They are capable of self-renewal. They can renew if some cells are damaged or old, and they are removed and replaced by another new cell by the stem cells. So self-renewal, and they exhibit cellular potency. They have the potentiality to divide. What are the cells? They have the potentiality or capability to divide and produce. That multiplication potentiality also they possess. So these stem cells, they can differentiate into all the types of cells. If you are placing the human stem cells in the pancreas, and they can develop into pancreatic beta cells or alpha cells. If you are placing in the damaged liver cells, the damaged liver cells are removed and the new stem cells can develop into liver cells or hepatic cells. So this is called stem cell therapy. Okay, so there are two types of stem cell therapy. One is embryonic stem cells, another one is adult stem cells. Right? Embryonic stem cells, shortly called as ES cells, they are pluripotent in nature. Embryonic stem cells are pluripotent in nature. That we'll discuss next. Pluripotency means the stem cells they can differentiate into any of the three primary germ layers. We have studied ectoderm, mesoderm, endoderm. Like this, pluripotent means they can produce any three primary germ layers. From the ectoderm, the skin and nervous system will develop. Like this, embryonic stem cells are pluripotent. They can develop into three primary germ layers, and it is isolated from the epiblast tissue of the inner cell mass of the blastocyst. Blastula stage of the embryo. I'll show the picture next. In the blastula stage, the outer mass as well as inner cell mass and the inner cell mass of the blastocyst and adult stem cells, they are found in various tissues of the children as well as adults. It is present. So first, if it is taken from the embryonic cells, then it is called as embryonic stem cells. Adult stem cells, right? They can divide and create another cell similar to it. So they are having the cellular potency, so they can divide like mitotically and create another cell similar to the adult stem cells. They are multipotent. Multipotent, they can differentiate into various kinds of cells that are related. For example, blood cells, blood stem cells, they can differentiate into any kind of the blood cells, lymphocyte, monocyte, neutrophil, platelets like this. So by this multipotentiality of the adult stem cells, they can act as repair system of the body. So all the damage of the body can be repaired by means of the adult stem cells. So our red bone marrow, we have studied bone marrow consists of yellow bone marrow and red bone marrow, and this red bone marrow is a rich source of adult stem cells. So this is very useful for treatment, okay, and applications or uses. So we can produce more kinds of generation of production of new stem cells and the tissues. It could be used for cell-based therapies. So the treatment, whatever the treatment, we can use this for various treatment techniques for curing many cancer and various diseases. We can use this technique of stem cell therapy. It is also used to test various new drugs, how they are applicable to this new patients. Like this, we can test the new drugs, whether we can use or not, its side effects like this we can discuss. Stem cells are nothing but undifferentiated cells found in most of the multicellular animals. They are capable of self-renewal as well as exhibit cellular potency. Now, what are the characteristics of the stem cells? Already we have discussed the same characteristic features of the stem cells in the point-wise and diagram-wise I'll give you. So, what are they? They are undifferentiated cells. They are not differentiated into any particular kind of cells. So, wherever it is uh, placed or transfer, they can develop into that particular kind of cells. Capability of self-renewal. They can capability of self-renewal, and they can produce its own cells again and again, multiply and produce self-renewal, and exhibit cellular potency. All the cells, they have the potentiality to divide mitotically, and the cellular potency they possess efficient to differentiate into other cells. When they are implanted into other cells, they start it's differentiated into that particular kind of cells. So if you are collecting the stem cells in the culture medium, it's culture, and it can develop into any cell. Wherever we are just placing, it can develop into that particular, differentiate into particular type of cell. For example, the same stem cells, if it is introduced in the nerve cells, it can develop into nerve cells. Placed in the blood cells like lymphocyte, monocyte, okay, and it is placed in liver, or they can develop into liver cell or hepatic cells. It is placed in the muscle, it can develop into muscle cells. So stem cells are undifferentiated cells only, but wherever it is placed, they can develop or they can differentiate into the same that particular kind of cells. They can differentiate. This capability is very useful in treatment of various diseases. Okay. Now, what are the source substances? I said human being or human body possesses various stem cells, and we'll see some source substances. Placenta, you can see placenta, umbilical cord, and placenta and umbilical cord. This is the umbilical cord which connects the fetus with the mother's uterus, and this is the placenta, umbilical cord, and this is the amniotic sac which is filled with the fluid called amnion, right? This is the amniotic sac which surrounds extra embryonic membrane surround the baby immediately, and here the baby is kept inside without any shock or anything, safely by amniotic fluid. It is filled here, and you can see the bone marrow. Which bone marrow? Red bone marrow is a rich source of stem cells. Okay, and yellow bone marrow is mainly consists of fatty tissue and blood. Blood also is a major source of stem cells, blood, and this is called the blastocyst stage. In the blastocyst stage, the outer mass of the cells are called epiblasts, and the inner mass of the cells also useful for as a source of stem cells. So we can see amniotic sac and amniotic fluid, placenta along with the umbilical cord. Umbilical cord, here is the placenta, and blood, and epiblast. Outer part is called epiblast of the embryo or inner cell mass, and what is it called? Red bone marrow of the, what is it called? Red bone marrow. All these are the main source of stem cells. Now, what are the different types of stem cells? Upma? The stem cells are two categories. One is embryonic stem cells, another one is adult stem cells. So I said embryonic stem cells, they are derived from blastocyst stage of the blastula, and that is the embryo. It is derived on which stage? Blastula stage, and from the blastocyst, the particular cells are derived, and they can develop into stem cells. It can be preserved. The embryonic stem cells are pluripotent. So what is the meaning for pluripotent? I said they are nothing but they can differentiate into any of the three germ layers, capable to differentiate into any of the three germ layers. That is ectoderm, mesoderm, endoderm. Here you can see the adult stem cells. So hematopoiesis means process of production of blood cells in the red bone marrow. It is called hematopoietic stem cells. It is an example for adult stem cells. It is multipotent. It can differentiate into various kinds of cells. So various kinds of cells. So here all they develop into three germ layers I said. Here they can develop into various type of cells. For example, this is RBC, WBC, platelets, like this muscle cell, nerve cell, like this they can able to differentiate. So adults, if it is hematopoietic stem cells, RBC, WBC, platelets. So normal, any other adult stem cells, they can differentiate to any kind of the cells. So adult stem cells are multipotent. They can develop into various kinds of cells capable to differentiate into any of the related cells. Related na hematopoietic blood related stem cells. So hematopoiesis is the process of production of blood cells, and in this RBC, WBC platelets can be produced. Now, I said some terms. I'll explain to you once again. Totipotency. Totinanapa, total. Ability of the single cell to divide and produce all the differentiated cells in an organism. Right? A single cell can divide and produce all the differentiated cells of an organism is called totipotency. From a single cell, the whole organism can be able to develop. Pluripotency, this is possible only in most of the plants, many plants. Pluripotency, plurina several. So a stem cell has the potentiality to differentiate into any three germ layer, ectoderm, endoderm, mesoderm. Multipotency, multina many. Stem cells can differentiate into various types of cells that are related. For example, blood stem cells differentiate into lymphocyte, monocyte, neutrophil. So blood stem cells are hematopoietic stem cells. So they can develop into lymphocyte, monocyte, neutrophil. All these are WBCs, and they can also differentiate. Blood stem cells can differentiate into RBC, platelets. Also they can develop. Right, oligopotency, oligo na few. So stem cells can differentiate in few cell types only. For example, lymphoid, myeloid stem cells can differentiate into B lymphocytes and T lymphocytes. They can develop, but not as RBC. So இது வந்து B lymphocyte T cells தான் develop ஆகும், not as RBC or red blood corpuscles. Unipotency refers to union single ability of the stem cells to differentiate into only one cell type. Can you understand now? Now, coming to this stem cell banking. What is the meaning of stem cell banking? So, we have collected the stem cells maybe from the red bone marrow, umbilical cord, or from the placenta or blood we have collected. The extraction of the stem cells. From that, we are just collecting and extracting the stem cells. So before usage, we have to process it in a proper way. Processing and storage of the stem cells for the future. என்ன பண்ணும்? Extraction, processing and storage of the stem cells for future use. So these are the three important steps in stem cell banking: extraction, processing method and storage as stem cell banking. So in the stem cell bank, you can see we can save our own body stem cells there for many years. And whenever that disease condition, we can use our own body stem cells for our own treatment. We can use. So what are the types of stem cell banks you can see normally? Amniotic cell bank, cord blood bank. So cord is umbilical cord. Amniotic cell bank, it preserves the amniotic fluid. And cord blood bank, it preserves umbilical cord. So at the time of childbirth, the umbilical cord, it is used as waste by most of the persons. They think like this, but it is a major source of stem cells. And the amniotic fluid and the amniotic membrane, they act as amniotic cell bank. We can preserve after processing for many years for the treatment of our own diseases. So cord blood, umbilical cord tissue, it can be processed and kept as storage for future use. For our own treatment, we can use this. So stem cell banking means extraction of the stem cells, processing, and we can store for our future use. Okay, and mostly the amniotic fluid, it is from that the blood it is taken means amniotic cell bank. Umbilical cord, if it is collected, it's called cord blood bank. The stem cell banking, in this, what are the three methods I said? Extraction, processing and storage of stem cells. So amniotic cell bank is a facility that stores the stem cells derived from amniotic fluid for future use. அதே கார்ட் பிளட் பேங்கிங் என்ன சொன்னேன்? கார்டுன்னு என்ன கார்டு? umbilical cord. Extraction of the stem cells from the umbilical cord during the childbirth. So at the time of childbirth, the umbilical cord is taken safely and it is kept, and stem cells are extracted and processed, and it is kept in the bank, and it can be useful for the mother or the baby in the future. Umbilical cord and cord blood are the most popular source of stem cells. The placenta, amniotic sac, amniotic fluid, all are rich sources in terms of both quantity and quality. So we have studied how the stem cells are useful for treatment of various diseases in our body. So these stem cells in the human body, it may be adult stem cells. If it is taken from the embryo, it is called as embryonic stem cells, and it can be preserved, extracted, preserved, and useful for various treatment of various diseases. Yes.